Hi everybody, I'm going to just go over the setup for the enzyme lab that you're going to watch me do. We have a bunch of things that we need to do this particular lab together. So I'm going to focus in and show you everything and explain what it is. I'm going to take the milk and add 20 milliliters of milk to both of my beakers. All right, these are little test beakers, so I can see how much I'm adding. Hold it up to eye level. For this experiment, it's not critical that I have exactly 20 milliliters. If it had been, I would use a graduated cylinder. All right, so I have my 20 milliliters of milk, room temperature, in both beakers. I've already taken one of my lactate tablets and crushed it. And so in this bowl, there's powdered lactate. Lactate is the brand name for the enzyme lactase. I will add the lactase to my test beaker and then start the timer. Once the timer's started, I'm going to wait about 5 minutes for the lactase to interact with the milk in the test beaker. During that time, I'm going to treat both beakers identically. I'm going to hold them to keep them warm, and I'm going to swirl them for 5 minutes. And after 5 minutes goes by, we're going to test both of them to see whether or not there's glucose in either of the beakers. Okay, so I'm going to put 2 spoonfuls in there. Start my timer. Nope, that didn't start it. Start my timer and start swirling away. So I'll just walk around and swirl for 5 minutes. So 5 minutes has gone by. And now we're going to test both beakers for the presence of glucose by dipping these glucose test strips into both of them. We dip it in for just a second, and then we wait about a minute and compare the color of the end indicator, which right now is a kind of pretty blue. It's going to change colors, and by looking at the color change on the index here on the little container, we'll be able to tell how much glucose is in both beakers. Ready? Here we go. Okay, so we're going to go dip, get them wet. Left hand is control, right hand is test. It's going to turn this sideways, and we're going to wait like a minute and see what happens to both of them. Hopefully they'll stop wiggling in that time too. I'm looking at the clock on the wall to time myself, and we'll just watch and see what happens. I think you can see that my control beaker glucose strip isn't really changing color. It's still blue, but my test beaker is getting darker and darker and turning green, and now it's getting a little bit brown. We're about 30 seconds in. A little bit longer to go before we take our final measurement. Whoops, I also made it start wiggling, sorry. 45 seconds. Oh, this is so exciting. Not really. 50 seconds. 55 seconds. And that's 1 minute. All right, so I think we can agree that the control beaker has no glucose in it. That's what this no change in color indicates. So no glucose here in the control beaker. However, our test beaker is somewhere between this dark green and this brownish green. So it has somewhere between 250 and 500 milligrams per deciliter of glucose mixed into the milk. That means that some of the lactose in the milk has started to be converted to glucose and galactose.